Good morning, good afternoon, good evening, wherever you are. This is Mrs. Casper coming to you with a lecture on anatomy and physiology. This is for my private students, and I'm very happy to have you. I will keep your name private, but I'm happy to have you along for the ride here. We'll be looking at anatomy and physiology. It is also known as Biology 2 or Advanced Biology. We're going to see here, this is what we're looking at, the book to your left there. This is put out by Apology of Science. It's an excellent book. This is a newer edition. If you have the older edition, the first edition, it actually is in the same order and everything. It'll work just fine. But this new one has better pictures and things like that. All right, so this is a course that is designed for high schoolers, mostly for juniors and seniors in high school, because it does cover some very complex ideas about biology, about the human body in particular. I will say, because all of us have a human body, it's very interesting that way, but you'll be absolutely blown away at how complex our human body is. It's crazy. And it will definitely lead you to, in my opinion, say that there is a designer at work here, okay? That it didn't just happen accidentally over millions of years. There's really no way for that to happen. Okay, the way I teach this class is a little bit different than other classes, and that's because I really like to keep the information in its context. And what I mean by that is that in a regular course, when I would teach this in a classroom, I would spend the entire first week of the module lecturing the whole time and students taking notes the whole time. And then the second week we got together, we would do nothing but a quiz and a lab. So you can see right here, there would be a lecture. Okay, in this particular lecture today, we're going to be reviewing Biology 1 mostly. And then we would do a lab right here. Now, because this is a remote class, we won't be doing that lab. I understand many of my students will indeed be able to do a lab elsewhere, which is good. Happy to hear that. But my point being is that you will get the entire module in one long lecture. Now, I'll tell you where it breaks to part 2, and you can pause it there and come back the next week if you'd like to take it a little slower. In fact, you can pause the video wherever you want to if you'd like to take some notes. You'll be reading the textbook. That will go along with the things I'm telling you in the lecture, but I don't believe you'll have a lab book or any kind of note-taking booklet to write down in. Now, if you write your own notes, that's great. It's helpful. And if you're doing lab experimentation type things, you'll need to write that down somewhere. So maybe get some sort of little blue book or something like that. If you do have a desire to do more lab type activities, let me know and I can tell you how to get a hold of some of those materials you'll need, or you can borrow some of the materials from me. If you like something along those lines, would be great. All right, so here's how this kind of works here. We'd start with our lectures. So we have modules 1 through 16. And then, like I said, it'll all be in one big lecture. Okay. All right, so this is called Anatomy and Physiology, or A&P. Okay, so if you mention to an undergraduate, that'd be a person in college, A&P, they might shudder a little bit, might shiver, because it is difficult. There are some difficult parts of it. But like I said, it's fascinating. And it's great stuff to learn. If you have any interest whatsoever in any kind of medical field, going into med school, nursing school, into sports medicine, into, massage therapy even, things like this, it's very good to know these things. Even veterinary science would benefit from learning anatomy and physiology, A&P. Okay, so let's get started on some background here. So the introduction, anatomy. What do I mean when I say anatomy? All right, anatomy is structure. That's the stuff the body's made of, okay? So we have the structure is basically the things we can see, even if we have to see them with a microscope. You see what I'm saying? So it's the stuff that is very visible to us. We would do some dissections, for instance, and then we would do some microscope work. So gross anatomy, gross means big, right? It's going to be the big stuff. It's macroscopic. I can see it with my eyes. I can touch it with my hands. Fine anatomy would be microscopic. I need the scope in order to see that, but I can still see it. So we're going to study this in a systematic anatomy. In other words, we're going to be looking at the systems of the body. There are 11 organ systems, and I will bring those up here in just a minute. The next picture I'm going to show you is a little disturbing, but it's just a model, so don't get too frightened. Here it is. It's gross, right? It's gross anatomy. So you can see all the big stuff we're looking at here, okay? So we see a pretend cadaver opened up here. Now, there are people that do donate their body to science so they can use a cadaver. If you go to med school, you will have a cadaver of a real person, a real dead person. You will have the same cadaver all year long, and you will dissect it throughout the year. So, but gross, remember, means big, so I can see the big things here. So here's a human body. We'll be looking at all those systems you can see right there. You can see some blood vessels, you can see some muscles, you can see some guts, lungs, heart, that kind of thing. All right, so the study of macroscopic structures of an organism is what you can see without a microscope. That's gross anatomy. Microscopic structures is microscopic anatomy, looking at it under the microscope. So you can see here different items under the scope. Right here, these cells are all touching each other, making me feel like they're epithelial type cells. We'll get to all of that. I will tell you that each one of these has been stained. Purple and red and another purple color. We do that so we can actually see them. If you look at actual tissue under the microscope without stain, it's very clear. It's clear. You see right through it. So it's hard to make out any kind of structures in there if we don't have a stain on it. Now, when you do stain a specimen, it does kill it. So if you had, for instance, a small creature from the pond or something and you wanted to stain it, you would have to kill it first to do that. So that's what staining does to my specimen. But then again, it also helps me see. There is a time when in biology we would study like pond water, for instance, and see little creatures swimming across, that kind of thing. We're not going to really cover that in biology too, except to say that, for instance, when we are looking at our digestive system, it's really great to have a nice healthy microbiome. And a microbiome is the amount of bacteria that are living in your gut. And which turns out it's very, very important for us, and that's the whole bacteria part. My degree is in microbiology, so I love bacteria and how the world has come around to, instead of being afraid and worried about bacteria, we know that it's our friend and we can use it to make us healthier, that kind of thing. All right, histology is the study of tissues, okay? I mean, there are people who have a degree in histology. They know all about the tissues of the body. It turns out we have 4 tissue types in the body. This is something that for a quiz you would need to know. You would have to write that down. It looks like we can see all those types up there at the top. We have epithelial, we have muscular, we have nervous tissue, and then we have down here what is known as connective tissue. And there's different types of connective tissue, and we will go through each one of those types in time. All right. Now, what about physiology? I'll get back to tissue, by the way. What is physiology? Now, this is the hard part of the course, okay? Physiology is the underlying mechanism, the function of the structure. If something is working in a certain way, for instance, the heart is beating, all right, we know that. We can talk about the chambers in the heart. That's all anatomy, looking at a heart, dissecting a heart, this kind of thing. Why is it beating? What's causing it to beat? That's the physiology part right there. It's the function and the mechanism of it. The picture you happen to be looking at here is kind of scary, and that's because it's something known as the sodium-potassium pump. We see sodium here going in. We see potassium. I guess the sodium's going out, the potassium's coming in. This is the way that our muscles contract, okay? And it goes through a process or a mechanism by which this happens, and it's very, very complex. Okay, and in biology too, in A&P class, you will have to learn about the physiology of the sodium-potassium pump. It will come in a later module, and it's in the muscular module, and it's fascinating. I'll play some videos that'll help you understand it. My goal here is to help this to be more accessible to you, that it's not just scary stuff, right? But it is one of those things that it's difficult to take a quiz on, right? So whether or not you're taking a quiz is up to your tutor, teacher, or parent, all right? So that's up to them. All right, part of the deal we are looking at in anatomy, remember that's looking at the structure of something, not the function necessarily, is we compare it to other living things. So here, comparative anatomy, we're going to see, if you look at the bones here, this is the bones of a human arm, okay? And notice the color of the bones are different to help us to point those out. Now let's compare it to a cat, All right, look at the humerus is a long bone. Look at the cat's humerus. Wow, it looks almost identical, a little shorter. Our radius and ulna right here in the forearm, right here, looks very similar there. We have our carpals and metacarpals. That's our wrist and the top of our hand. So does the cat. Different. They have a claw coming out. We have fingernails. Now, but check it out compared to the whale. The whale also has a humerus and a radius and ulna and carpals and metacarpals and phalanges. Okay, phalanges would be fingers and toes. And a bat, similar right there. So we see this that compares well. Some people will take this kind of thing and say, well, you see, we all came from a common ancestor. This is a proof of evolution. Well, for one thing, science proves nothing. And secondly, evolution's a theory. All theories can be overturned if there's enough evidence to overturn them. In my opinion, as a creation scientist, someone who believes that God created this and furthermore created humans as a special creation, I will say that God had a good design and he stuck with it. That's how I'll look at this right here. So that seems to make just as much sense as this evolution kind of a thing. All right, systemic or systematic anatomy is the study of the systems. So there are 11 organ systems. So this is something, if you're taking notes, you would write down these 11 systems. And if you need to stop the recording, you can do that. So here are let's get started on our 11 human systems. We have a chart. I'm going to have a chart for you here in a minute. This is just going to show you some pictures. The integumentary system is our skin, our hair, our nails, and the glands that are in our skin, sweat glands, things like that. The muscular system makes up the muscles, right? And that's the muscles, mostly we're talking what's called the skeletal muscle. In other words, muscles that are attached to bone versus like the heart, for instance, which is also a muscle. We have the skeletal system, all the bones right here. We will talk about the names of bones, where they're found. We have the nervous system. It includes the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which is all the nerves headed down to, every part of your body. The endocrine system is your hormone system, all the hormones in your body, starting here in your brain at the pituitary gland and the hypothalamus to the, thymus and thyroid. And then so many, like this is the pancreas right here, and it produces a very important hormone called insulin. We'll learn about that we have a whole module, a whole unit on the integumentary system and these other things. I think nervous system has at least 2. And skeletal muscular has, between the 2 of them, there are 3 different modules there on that one. Circulatory system, that's the heart and the vessels and blood. All right, then come lymphatic system. That's the secret hero of our bodies. Basically, it is the immune system. It's what helps keep us well. It's very interesting, actually, because it goes all, see all those little vessels going through the body here, just like, it looks like veins, but they're not. And they don't have a heart to pump everything around. It's very interesting. We'll find out about that. Respiratory system, that's breathing and lungs. It sounds like it might be simple, but I will say, in my opinion, the most difficult module in this whole course is Module 14, which is learning about the respiratory system. And that's because we have to apply something we learned in Biology 1, and what that was is called the Krebs cycle, right? The Krebs cycle. Scary stuff, right? It's cellular respiration. Cellular respiration is complex, but also super fascinating. All right, digestive system, well, we talk about eating food and how it gets digested in your gut. And then we have some accessory organs that help digest, like the liver and the pancreas there as well. Excretory system would be the urinary system. That's the kidneys and bladder. And then the very last module, Module 16, I call it the dreaded Module 16, is all about the reproductive system. We'll get there eventually. All right, the structure of the body, macroscopic and microscopic, here we have. So we start with the whole organism. That would be the human body, okay? There's the whole organism. And we can look at it by way of its organs. So that'd be the organs that are the organ systems. And the organs are made up of tissues, and the tissues are made up of cells. The cells are made up of organelles within the cells because we are eukaryotic creatures. And the organelles are made up of molecules, and the molecules are made up of atoms. All right, so you can see from the very small and submicroscopic here, we can't see atoms and molecules with a microscope. We can see organelles, cells, tissues, and we don't even need a microscope to see an organ. An organ, for instance, an example of an organ would be your heart. That's an organ. And then the organism is the whole human right there. All right, so the organs are collections of specialized tissues. See the word tissue there, right? Which perform specific functions. All right, so we have the brain as an organ right there. The eyeball is an organ. These are both part of the nervous system, and so is the ear. Okay, because the ear for hearing, that uses the nervous system. We also have right here, there's the hearing part of the ear. Right here is the other sense in the ear, which is the sense of balance. The heart is an organ. I think right here they might be talking about, I don't know if they're talking about the skin or the bones is an organ. These are systems now, but this is an organ. The skin is the largest organ in your body, in fact, is the skin. The stomach is an organ, and this is showing a baby, so that's looking at, I don't know if we call that an organ so much as a system, which is a reproductive system. All right, so back to tissue. I told you I'd come back to tissue. There are 4 types of tissue. You need to remember the 4 types of tissue for the rest of your life, okay? These are the 4 things. Like the 4 Gospels in the Bible, we have the 4 types of tissue. All of these fascinating cells and organs in our bodies can all be boiled down to 4 different types of tissue or combinations of those tissues. All right, collection of specialized cells. There are 4 types. Number 1, epithelial tissue, right here. Epithelial cells. That would include skin and membranes, the inside of your mouth, like that, the inside of the linings of your body, and that kind of thing. Then we have connective tissue right here. Like it says, it connects things together. It holds things together. That would include bone. It would also include blood, interestingly. Muscular tissue right here. We have muscles, right? Those are the muscles, those little stripes in there. We're going to learn why they're striped like that. And then we, of course, have nervous tissue. Nervous tissue here would be, these are the types of cells. They're crazy-looking cells right here called neurons that send electrical signals down there. And they mostly send the electrical signal to other nervous tissue or to muscular tissue. So muscles can move. That's how they move. So those are the 4 types. I would write those down. And you will be, if you get a chance, looking at, in our, I think, Module 2, we'll actually talk about the tissue types, and I'll have some microscopic slides on the presentation, and hopefully you have a scope to look through. All right, what about the physiology? Remember, this is the hard part. So it's a mechanism or the function of the system. The idea here is we're going to maintain our bodies. Okay, our bodies need to stay the same as much as possible. We call that homeostasis. It's really about balance. And the balance word that is used in science is called equilibrium. Equilibrium just means everything's equal, everything's balanced, okay? We want to make sure that our bodies don't get overly stressed. It's a constant battle against stress. So the state of equilibrium is controlled by our brain, but also very heavily by our hormones. And keep in mind, the hormones are controlled by the brain too. So the brain is, of course, the mastermind, if you will, but the hormones come in a close second. What we're looking at over here to the right, this little fancy picture right here, which is an artist's idea, this is actually a nerve cell right here. This is called a synapse. That's the gap between one nerve cell and the other. So think of that kind of like an electrical cord that's coming in and plugging into the wall. It doesn't actually ever touch it. Isn't that interesting? Instead of touching it, what it does is it sends little chemicals right across here called neurotransmitters across that little gap right there, and then causes the electrical signal to continue down to do whatever it needs to do, move a muscle or something like that. So the idea that we're under stress, let's think about that for a minute. What stresses your body out? I'm not talking about, oh, I'm nervous, I have a test coming in, I'm stressed out. No, that's not it. The stress we're talking about here is actually a physical stress. For instance, if you are outside in the Texas area right now, this August, right? It's really hot. It gets hot smack in the middle of the day. And so what does your body do to try to deal with the heat? Well, you start to sweat. Right? So when you sweat, what happens? The air blows across you and the sweat evaporates and it cools you down. You also kind of turn red, right? You know why you're turning red? When your skin gets red? It's because blood is coming to the surface so it can cool you down, so it can have access to the wind that's blowing across you. You know, right now, for instance, I have a little fan on because even though I'm inside with air conditioning, thank you, Lord, for that, it's nice to have the air moving, because when the air is moving, it actually can, you know, cause that evaporation of any kind of sweat or something like that. Okay, so that's a way of handling stress. There are other stressful things that come into our body. Getting cold is another one, you know, but we'll go into that when we get to the nervous system. So here we go, homeostasis. So balance it out between stress. So homeostasis is the state of dynamic equilibrium. Dynamic meaning always changing, equilibrium, let's keep it the same. So dynamic, changing, equilibrium, keep the same, right? We're trying to get a balance there. It's another way of saying balance. The body is in respect to its internal environment and its functions and its external environment, really. The set point is the ideal normal value of a variable around which homeostasis is maintained through a normal range of values that are acceptable to the body. Okay, that's a lot of words to say. Our body is, like the temperature of our body is about 98.6, right? That's general. Every individual can have a little bit of a different body temperature. But we have a set point. We want to be at a certain level. If somebody comes to the doctor and they have a temperature that's 104, ouch, that's way out of the set point. So that needs to be addressed. If somebody falls into a frozen pond and they get very, very cold, that's very stressful on the body as well. So therefore, we need to deal with getting them back to a regular temperature, bringing them back to homeostasis. By the way, a fever is actually good for us. It helps. The reason why our body does that is it heats up so that bacteria can't grow very well. How about that? God designed it beautifully. And the stress is a factor that causes one or more physiological variables to move away from that set point. I'm too cold, I'm too hot. Even things like, hunger, things like that. We're going to do something to satisfy the stress of hunger. All right, so if you wanted to write these down, that's okay. There's a lot of words to explain those things. It's all in your book, written down, I think, word for word. And so if you need to stop this and write some of that down, you're free to do that. All right, what's an effector? That's a structure in the body that can change the value of a variable. For instance, remember we talked about, I guess it was a different class, the blood pressure idea. If I have a certain amount, measurement of blood pressure in my body, okay, and there's a certain range that's considered healthy for a human to have their blood pressure. You may know somebody who has high blood pressure, okay, and if somebody has high blood pressure, it puts a lot of stress onto the blood vessels, right? That it's actually the blood is slamming up against the side of the blood vessels, and it's stressful on that. So we want to keep that blood pressure down to a regular normal place. Some people have low blood pressure and sometimes they feel kind of a little faint or something like that. But the way our body deals with it is through a feedback system. All right, we have positive feedback systems and negative feedback systems. The most commonly one found in the human body would be a negative feedback system. Okay, we're going to battle stress. What happens in a negative feedback system is that we detect that there's a change, something's wrong, and we produce the opposite thing to equalize it, to bring back the status quo or homeostasis. So let's look here. So then let's go to negative feedback first. So my body temperature rises. I'm outside in the Texas heat. My body temperature goes up. So what do? I sweat, and then the wind blows, and my body temperature drops because I'm cooling off. Okay? So the idea here is that my body's doing something to mitigate or to bring back into balance the thing that's a problem, right? Now, there is such a thing as positive feedback. We see here it's talking about a bank account where my account balance in the bank grows, I get more interest, and I get more, and it keeps going and going and going. It feeds upon itself. In the human body, you will find it now and then, mostly when it comes to reproduction and producing a baby, honestly, is where there's a positive feedback to keep it growing, kind of like your money growing in a bank account. All right, we're going to do a little review here. It says to draw this in your lab manual. I don't know if your folks want you or your tutor would want you to draw this. But remember, in Biology 1, you went all the way through. You probably made a model of a cell, either with clay or Jell-O or something like that. I believe my students used a giant cookie, if you remember using a giant cookie and then putting the icing on there and then the different types of candy on there, which would be a fun way to remember the parts of the cell. So just review that in your book, read about it. Namely, we have the nucleus reasonably in the middle of the cell. Doesn't have to always be in the middle, but inside the nucleus, one thing you need to always remember is that the DNA is in there, okay? So DNA is always in the nucleus of the cell. It always stays in the nucleus of the cell. It doesn't come out. The things that come out of there, we're going to see that in protein synthesis, RNA comes out. So DNA stands for deoxyribonucleic acid. And it's the code that codes for everything, codes for you, your individual traits, what you look like, how tall you are, that kind of thing. But it also codes for everyday living, how you're going to make your proteins, how you're going to make your energy for your body, that kind of thing. Okay, but here a couple other things. We have our endoplasmic reticulum, some of it rough because it has ribosomes on it. Some of it smooth because it doesn't have ribosomes on it. Ribosomes are the little tiny dots there. That's where protein synthesis takes place. It's a very important part of the body. Mitochondria, you know that. Say it with me. It's the powerhouse of the cell. That's right. That's where the ATP is made. So I've told people oftentimes that in biology too, oftentimes when I'm asking a question, the answer is going to be ATP. Okay, what is ATP? It stands for adenosine triphosphate. It is it's a chemical that's being made in your body. It has very specialized cell bonds in it, and when those bonds break, they release a lot of energy. So it's the energy of our body. Life is about energy. All of life is about energy. Where are we going to get our energy, right? So in your body, where do you get your energy? From your food, right? For sure. But also just think about economics. It's all about energy. Where do we get our energy? Well, from gasoline that we put in our cars and our planes, that kind of thing. I don't know if you pay attention to any politics or anything, but a lot of it has to do with energy. Where are we going to get energy from? And so our cells are in the same way. They want to get their energy, and they're going to take that and convert it into the energy that's used by the cell. Okay, so read that in your book and just review the cell and the parts that are in it. We'll go into some depth on some of these. For instance, we'll talk a lot about the mitochondria when we get to cellular respiration. We'll talk about the ribosomes when we get to protein synthesis. Let's see what we've got here.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
All right, so that was a review of cells. How about that? We're going to go a little more into there. So remember, the nucleus contains the DNA, right? Deoxyribonucleic acid. You can see here it's floating around in there. It doesn't really move around necessarily like that. But this is the endoplasmic reticulum under the parts of that. Okay. The mitochondria, it's a big deal. So we're inside, this is actually a really enlarged picture of a mitochondria right here. See all those little folds inside there? You'll find this often in the human body and in other tissues, that the folds actually increase the surface area, where there's a lot of area there for whatever is the function to take place, all right? So it's known as the powerhouse of the cell. It's where adenosine triphosphate, or ATP, is synthesized. That's the stuff we use for energy. If the fundamental chemical needed by the human body to do work. There is a little bit of mitochondrial DNA. I'm going to show you, I think in the next slide, you're going to see there's a special bit of mitochondria in a little ring. You get it from your mother. It codes for most of its own proteins by itself. Here's a picture of it. Okay, so you see this little ring right here like that. There's these little rings. See that DNA that's inside there? It's inside the mitochondria. You get it. So most of our DNA is in here. That's controlling our body right here. But for energy, it's right here. It may be really important to the future of understanding genetics a little better. The reason I say it's very exciting, but the reason I say be careful is because one of the theories, going around out there is that this mitochondria itself used to be a little microorganism that had its own DNA, could have its own life outside of the cell, and that one of our big cells like engulfed it. We'll talk about engulfing and how cells can, quote unquote, eat things, a long time ago, and it was part of the evolutionary process. I don't believe that. I believe that it was created whole inside these eukaryotic cells, so that, because how could that cell get energy without it? All right, so if there's a million years going from this time to this time, how did the cell get energy, right? So it doesn't really make sense evolutionarily. But that is so bizarre how they have these little rings of mitochondrial DNA. They're still doing a whole lot of research on there. The reason why it all comes from your mother is because during the process of fertilization, when the sperm meets the egg, all of the mitochondria have to do all the work to get the sperm inside the egg, and in the process they get destroyed. So therefore, every bit of mitochondrial DNA you get has to come from your mom, because the stuff that was contributed by your dad was destroyed in the process of fertilization. Very interesting stuff. It's really interesting to look back that way. They say that it's easier to follow the DNA, in the sense of somebody getting their DNA tested or whatever to see what country they came from. It would be on your mom's side, because when it comes to for the mitochondrial DNA, it would be that way. All right, our endoplasmic reticulum, here's an artist idea, right? The smooth has no ribosomes, the rough has the ribosomes on there. It's in there to help with synthesis and things like making things. So this is a chart that in the One Day Academy lab book, this chart would be in there, and you would write these things down, okay? So these are the main things to remember about cells and the parts of the cells. So if you want to write this chart down yourself or take notes about this chart, you can. We've talked about these things. I'm going to get a little bit more about the chromatin is inside the where the DNA is inside the nucleus, but I'm going to talk a little bit more about the plasma membrane because it's so fascinating, and some of these other things right here as well. So if you want to write this down, you can pause the deal. Okay, which brings us to the second half of the lecture. So if you want to stop here for this week and then pick up part B next week, you can. But like I said, I like to keep these in context by continuing on, okay? So this is where One Day Academy would have split it up. All right, have a little video here, also review.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
All right, wow, there you go. Do you remember that from Biology 1? You you would have learned that in Biology 1, and in my classes I used to have these little codes, and the codes would code for a word, and then the word, they put the words together, make a sentence. It was kind of fun to do that way. All right, so the 2 steps of protein synthesis, this is review from Biology 1, is transcription and translation. So in transcription, it's copied, the DNA is copied by messenger RNA. So DNA is deoxyribonucleic acid, RNA is ribonucleic acid. Then comes the translation part, to translate what it is. It's kind of getting the mirror copy, if you will. In the ribosomes, it happens. The amino acids are arranged in a specific order to code for a specific protein by transfer RNA. If you need to review that, you can go back to your biology book from year 1 biology. All right, cell division, mitosis. Okay, cell division is called mitosis. It has 4 stages. Prophase. This is what all cells are sitting in, and if they're not dividing, and then all of a sudden the chromosomes will become visible. Centrioles, I think we'll have a little video for this one too, will migrate to opposite poles. Then we have metaphase. Chromosomes line up at the center. They connect to a spindle fiber at the centromere. That's the center part. Then we have anaphase, what we call the sister chromatids separate and pull apart. And then telophase or telophase if you're from Canada. Chromosomes gather at the opposite end. New nuclear membranes form around 2 regions. Spindle fibers disappear. Okay, so once again, this is review, but you probably remember that I would always say it as PMAT, so prophase, metaphase, anaphase, telophase. So we start with the mother cell right here. We have just regular DNA in there. These are the chromosomes. Okay, when they get all fat, we call them the chromatin. They fatten up because they're getting ready to divide. They duplicate. So this one duplicates, and it looks like a little X shape right there. They're in the middle. They're attached by the centromere. So you can see they're in that X shape. So they get here, and notice what's going to happen is they're going to pull them apart. So I'll have an orange one going that way, and an orange one going that way, a blue one going that way, and a blue one going that way, and I end up with the diploid 2 daughter cells that look exactly like the mother cell. This is a cloning process here. This is happening in our skin right this minute. It's happening in a lot of our cells, our bone cells and things like this. Especially if you have an injury, it's going to happen much quicker that way. But we're going to try to copy mother cell into 2 daughter cells. So what do we get in mitosis? 2 cells from 1. That's called mitosis. It's regular cellular division. All right, let's talk about how the details of that take place. This will be new information not covered in Biology 1. After telophase, that's the last phase, you end up with, let's go back for a second, you end up kind of here at telophase, right? And notice how I have these 2 kind of sides of it. And you can see here the 2 different sides of it. So I want to pinch that off, right? So after telophase, the complete copies of DNA are on either side of the cell. And the cell membrane then pinches off to divide the cytoplasm in half. It results in 2 individual cells. Okay, so the anatomy portion is I've got 2 cells. The physiology portion is there is a mechanism by which this happens. Okay, and it happens kind of complex here. We have a contractile ring that causes this furrow to form. Okay, and then it's going to slice that off right there. That's cytokinesis. I think I have another picture here. Yeah. So here's our telophase. I have my furrow, and then it's essentially just going to stretch apart that contractile ring of microfilaments. And if you remember from a previous video and early in this lecture, the microfilaments are made of a protein. So the protein had to be made by the cell in the ribosomes, right? All that had to happen before this cell division could take place. All right, I love the plasma membrane. It's a miracle of the body, of cells, even cells that are in, plants and things like this, less complex, if you will, have this plasma membrane. We call it a plasma membrane, but it has a couple of functions, all right? So the functions are, number 1, it delimits the cell. In other words, it helps the cell keep its shape. Some cells have to have a certain shape in order to function properly, okay? Like I think of a neuron. It has that crazy shape with the long arm coming off of it, and that long arm is important for an electrical signal like a cord to transfer down. 2, it provides receptors for sensing the environment. That's for protection and for transport. And 3, it exhibits selective permeability. So the receptors that allow things in and out of the cell will be selected by the cell. Not everything can go in, not everything can come out. It's going to be a protective measure here. So let's take a look at the plasma membrane.
All cells have an outer membrane called a plasma membrane, which acts as a boundary between the outside and inside of a cell. Cell membranes are primarily made of phospholipids, which are large lipid molecules, similar to triglycerides. Like triglycerides, they contain glycerol and fatty acids, but they differ from triglycerides in that they have a phosphate group, rather than a fatty acid, attached to the third carbon of glycerol. The phosphate head of the molecule is hydrophilic, or water-loving. This means that it interacts well with water. The fatty acid tails, however, are hydrophobic, or water-hating, and do not interact well with water. Because of these unique properties, when placed in a liquid environment, phospholipids tend to arrange themselves into a phospholipid bilayer, so that only the hydrophilic heads interact with a watery environment, and the hydrophobic tails crowd inward, away from the water. The plasma membrane of a cell also contains other molecules that aid in its function. There are a number of different proteins embedded in a membrane, and they serve a variety of functions. Some are involved in the transport of substances across the membrane, while others help in cell-to-cell communication. Short chains of sugars are attached to the outer surface of some of these proteins, forming glycoproteins, which serve as recognition signals to other cells. Cholesterol is present in the membrane as well. Cholesterol lends support to the membrane and regulates fluidity, giving the membrane the general consistency of olive oil. The phospholipids, proteins, glycoproteins, and cholesterol are all present in the membrane as a flexible pattern, commonly referred to as the fluid mosaic model. Together, these components of the cell membrane allow the cell to interact with its external environment.
Okay, there we go. I have some pictures as well. So the structure of it, you notice here, the hydrophilic, water-loving will be in the water environment on either side, and then these little tails are the hydrophobic ones, okay? Some of the things hanging off the top, just kind of attached to the outside, some of those are going to be specific to you. I don't know if you've ever heard of anybody who had a kidney transplant or something like that. They had to find a match that would match their system. And that is part of it right there. What's on the outside of their cells? That's how it matches, right? So the outside of some of our blood cells may have little indicators on there to tell you what type of blood you have. Type A, type B, type O, that kind of thing. Notice there's also these things going there. This looks like a little tunnel. So something can kind of be transferred right through this little tunnel right here. Other ones might allow something to come through in a more complex way. Here's one that has to fit exactly inside this little transport system. This might be just a marker right here because it comes out onto the surface and into the surface, or it could be as transport as well. So the idea here would be that we could get things in and out of the cell. Sometimes that goes passively. In other words, it passes right through easily, like water would go through. Other times it needs active transport. Okay, so active transport requires a little more mechanism there. All right, this is somebody, an artist's rendition. I've always liked this. It kind of reminds me of a sandwich, So we have right here the top piece of bread and the bottom piece of bread, and here's the middle part of the sandwich, but we have stuff poking all the way through. And then we have stuff that's just hanging off the end, right? That tells you, it's your cell. This one kind of goes through like this, an unusual kind of way to get through there. It is called the phospholipid bilayer because it's one layer and two layer, and it's a phospho and lipid. Remember, lipid means fat. So that's the fatty part right here, which would also be the hydrophobic area. The phosphate part would be attached to this top part right here, which is water-loving, or the hydrophilic area. Here's one. It's kind of showing how it's opening up, right? And you can see a little better here. This little protein would have gone all the way through and attached to whatever that is, probably to get something in there, something small. Here maybe something a little larger to get through this little tunnel area right here. And here a close-up again. I just am fascinated with it. That's why I like to keep showing you pictures of it. Okay, so how do they travel? How does the substance go? This is that hydrophilic head and that hydrophobic tail right here. Okay, so see how complex it is? We draw it like this, but it really has all this complexity to it. Another evidence right there for design, right? So hydrophilic is the water-loving part. Phil means to love, so I don't know if I have a son named Philip, and Philip means lover of horses somehow. Phobic, phobia, to be afraid of something, water fearing or water hating. A phospholipid is a lipid where one of the fatty acids has been replaced by the phosphate group. We saw that in the video. It's an important structure for the plasma membrane, mostly like for protection, also for the shape. We mentioned that as well. All right, what about selective permeability? What is it? It is the cell is going to have control over what comes in and what goes out, okay? So it's a protective measure, but it's also a mechanism for the homeostasis, right? We're going to have enough water in our cells. We're going to have enough chemical of certain kind in our cells. We're going to kind of keep big things out. In order to get big things out, I'm going to have to keep big things out. I'm going to have to decide whether I want them to come in or not. So if you look at this little picture right here, this is a barrier right here. It's just a solid barrier. Nothing can get across. But if I have a semi-permeable barrier right here, you see some small things can get through those little holes, but these big things cannot get through it. Now in Biology 1, you may have used something called a what do we call it? A dialysis tubing, and you put it into some water and you could see stuff travel in and out. I always do that for this class as a lab. It's really great. You just put a certain liquid with a color inside of a little bag, and then it goes into like a beaker or something, and then afterwards you can see how some of it traveled across, okay? But not all of it. If something's really, really big, it can't transfer. The only way I can get something really big into the cell is to use something known as active transport. Right here, this going through this little hole, that's passive. In other words, it fits. It goes through it, and then it will maintain a homeostasis when it goes through it. It will achieve balance. For instance, you can see here these little brown dots right here. Notice when it passes through, I have 1, 2, 3, 4, 5 on this side, and 1, 2, 3, 4, 5 on this side. I'm balanced now. It's not gonna, no more is gonna travel over there because it has enough. You get it? Yeah, you probably remember that from Biology 1 as well. So here we go. Some specific non-lipid soluble molecules or ions are gonna go through this little tunnel. It may be, it's called a membrane channel or channel membrane. It may have a charge associated with it. In other words, it might be a negatively charged channel which would attract positively charged ions to go through. All right, some things can't get through. They're going to bounce off. Some things are, this lipid molecule looks like it can squeeze through, and it loves to be here because it's in the lipid area, the fat area. So just depending on what the substance is, we'll see how it travels across. So transport through the membrane, we can see a little transport thing going on here, and something that looks like water just going through, right? That's H2O. H2O is going to, we definitely want our cells to easily be able to get water into them and out of it. So passive transport means I don't need any energy to get it through. It's just going to go through. If it needs water on the inside, the water will go through to the inside. We call that diffusion, right? You probably remember diffusion from Biology 1. The movement of molecules from an area of high concentration to an area of low concentration. Then it's going to even out on each side. We're going to have homeostasis. Active transport needs a special ingredient. What does it need, folks? What's the answer in Biology 2? Just like you go to Sunday school, the answer is always Jesus. What's the answer in Biology 2? It is, ready, here we go. Oops, what did I do? It is, there it is, ATP. We got to have ATP. We got to have energy. All right, so active transport requires energy. All right, and in order for it to work, there are 3 criteria that need to be fulfilled. There has to be specificity. Do they fit exactly in? Competition, is there too much going in at once? Do we need to slow down? That can happen sometimes. People with diabetes, for instance, have their glucose channels are all filled up and they can't get enough glucose in their cells. And that also can happen with saturation. So competition usually is different things trying to get in the same door, if you will. Saturation, there's not enough doors. Okay, it's all full, if you will. So let's watch this. Okay, here we go. So we have notice our, this is going to be active transport. We notice our phospholipid bilayer. Okay, now this is one of our little channels for things to get into. This is going to be active transport. This little item you're going to see is going to fit directly into this little spot in here. Then we have, boom, you saw that little like lightning bolt come in there. That's the ATP. I thought it started over by itself. I'm going to back it up and start over again. Okay, but let's finish explaining it. Fits there. ATP opens it so it can get inside the cell. So let's start here. Oh, watch it go in now. See, energy is ATP. Energy, boom, opens the door. Opens the door. So it's specific, and we have energy that pushes it in. Okay? There we go. Now, competition, we're going to see here, if I have the site in which everything is going to enter in, and here's an enzyme, okay, I can have an inhibitor that's taking up the spot. In other words, it's going to fit in there, but it's not what we wanted it to fit in there. And so the natural one comes in and can't do it. Sometimes this is used in medication to fill up a active site. For instance, if you have pain, it can fill up the pain receptor so that we can't get the pain anymore. So it's interesting to look at it in a medical sense that way. You can try to solve a problem by competitively inhibiting the active site. All right, active transport, if we got to get something large inside, okay, if something bigger than water, for instance, needs to get in, a large protein needs to get in, we can get those into the cell by way of endocytosis. Here's the word, endocytosis. We're going to get it into the cell, okay? We have two ways of doing that, two major ways. These are the big ways, actually, okay? So pinocytosis, we call that cell drinking. It's kind of absorb into, if you will. We'll watch a little video. And phagocytosis, very famous, cell eating, where it actually engulfs something. Think about, like in biology, when you learned how bacteria do that, how amoeba, will go and engulf something. That's called phagocytosis. Exocytosis is to get it out of the cell. So think about it. If my pancreas cells are making insulin, I need to get the insulin out of the pancreas so it can go to the rest of the body. All right, that's the job of the Golgi apparatus. The Golgi body is going to take that, it's going to pinch it off into a little vesicle, and it's going to send it out of the cell, okay? And we call that secretion. So that's a way to get things out of the cell. Other things are being made inside your cells as well, so glucose being one of those as well. Okay, so this, I believe we're looking at pinocytosis. It comes here, it gets enveloped, and it pinches off. So if you want to think about that cytosis part of it, it pinches off a little bit of the phospholipid bilayer. Notice it becomes in there like that, but it gets inside the cell. That's the goal. So here's phagocytosis. Notice that cell just kind of ate up that little bacteria. So what we're looking at right here, it engulfs the prey. This big purpley thing is a white blood cell. The little purpley thing is a bacterium, and the white cell comes in and engulfs it, eats it up, and gets rid of it, and saves us from getting sick. Okay, that's how phagocytosis would work inside the human body. All right, what do we have here? Let's see if I had a video. Yeah, I have a video. Here we go.
This microscopic video shows a white blood cell attacking a yeast cell. The particular species of yeast here is Candida albicans, which is a pathogen, so the white blood cell is eliminating it. This particular white blood cell is called a macrophage, and it eliminates pathogens by phagocytosis. In phagocytosis, the cell simply engulfs what it is trying to attack. As you can see in the video, the white blood cell has engulfed the yeast cell, and it will now simply digest the yeast cell to get rid of it. White blood cells, like this macrophage, engage in phagocytosis in order to protect the body. Other cells simply engage in phagocytosis to eat. Amoeba, for example, engulf their prey so that they can eat them.
All right. Pinocytosis is about the size. Remember cell drinking? I showed that video to you earlier. Here it can go inside. Phagocytosis is something big. Pinocytosis is something smaller. So this is says cell eating, and this is cell drinking, if you will. I think I have a couple of videos on these.
Pinocytosis, often referred to as cell drinking, is a way for a cell to take in substances that are too large to travel through the plasma membrane. When the cell encounters such a substance, it actually breaks its plasma membrane, allowing the substance into the cell. A pinocytic vesicle then forms around the substance so that it can be transported to wherever the cell needs it.
All right, and then we have phagocytosis. Oh, I thought I had a video on that one too. Let's see. Let me see if this is the same video.
Pinocytosis.
All right, sorry about that. I thought I had one on phagocytosis as well. Okay. Secretion, here we go. We're going to see it's being made in the cell somewhere. The Golgi body is going to take, looks like bubble gum, right? It's going to take it and send it out of the cell, okay? We see other transport vesicles here that are sending out smaller things. Secretions, we call those. Here's a regulated secretion. It's the process of getting material out of the cell via the Golgi body. Cells make necessary chemicals that other cells need. Example, I said before, insulin is made by specialized cells in the pancreas. but all cells need it, so it's got to get it out of there. And sometimes you can see it this way. The secretory vesicle will come up to an area, a pore, if you will, porosome. It's just a little area dip there that it can get to and therefore fuse right here and secrete that stuff right out. Why is the strict control of cellular transport so important? Well, the plasma membrane is selective and protective. It's the security system of the cell. So let's look at this one once again. So it let that first thing in, but it's not letting this in. It's too big. It's not going to let that thing in. Here comes something smaller. It can actually fit through the, there we go, going right through that. This one, nope, nope, nope. Wait a minute, there it is. Yay, now it can go inside. But we're going to keep that one out. So you see it's going to allow things. And what if that big thing right there was some sort of bacterium or something? That's how big it would be, actually. Pretty small, because we're looking at the side of a cell right here. So this is security to keep the bad things out and let the good things in. All right, so this is the end of the lecture. This is the end of Module 1. It was mostly review. And next week you would be taking a quiz and that kind of thing. But we're not doing that for this class, so you just need to take those notes. All right, I hope you enjoyed that. We'll get into some new stuff next time. And one thing I did want to mention to you is that sometimes you'll be getting a lecture that has been recorded recently, like here in 2025, like this one. But some of the other ones I've already had recorded, and so they'll be sent to you. The only problem with those, you will hear classroom noise, maybe some questions being asked by students and stuff like that. If they're too annoying, I will re-record those ones. All right, well, good luck. I hope you enjoy that. This is an excellent, excellent course. I just love it so much to learn about all the cool stuff. All right, thanks so much. So long.